Alright, my name is Mark Noonan, and today we will be looking at the interactions between prokaryotic and eukaryotic cells. Alright, so this will be my little PowerPoint on the prokaryotic and eukaryotic interactions. So the first part we're going to look over is the gut microbiome. It is one of the most important interactions between bacteria and the human body. It's a positive interaction. A lot of times when we think of bacteria, we only think of the negative parasitic relationship that we have with them, but the bacteria in our gut often provide a positive influence on our digestive health and our immune system. So the first article I looked at was titled, let me try and pronounce this, Bacteroids Thetaeomicron in the Gut, which I will shorten as BT, which BT plays an important role in the mucosal barrier reinforcement, which is on the epithelial lining of your digestive tract, immune system regulation, and the metabolism of nutrients. BT is a gram-negative anaerobe. And so the first part, in its mucosal barrier reinforcement, it ensures that only favorable substances are absorbed through the intestinal lining. That is what they found in this article. During the digestive process, that's really important to make sure that we are only absorbing the nutrients that we need and getting rid of all the other ones. I was also able to reverse the epithelial damage caused by inflammatory cytokines. One of the main ones I recognized was the tumor necrosis factor, which was able to reverse the damage of that. It also helps animals digest polysaccharides, which they would otherwise be unable to. In the study, it shows that there is that the animals required about 30% less caloric intake compared to ones that were gram-free. So the organisms that contain these bacteria do not actually have to consume as much because the bacteria plays a role in helping them obtain those nutrients. And to go along with that, it also helps with vitamin production. The second article that we looked at has the same bacteria, but also in addition to another one, which is called Lactobacillus johnsoni. And one of the important mechanisms of that was that it actually helped disrupt the fungal cell wall of the Candida species, which is a type of fungal infection, often causes yeast infections or vaginal yeast infections. They tested these in mice through oral administration. They used both of these bacteria, which reduced both size of the Candida species and of E. coli, which E. coli is a common, it's also common for foodborne illnesses. And they also recognized a relationship between these bacteria and certain disorders such as IBD, Crohn's disease, ulcerative colitis, and found that certain bacteria were reduced and certain bacteria were increased in patients with those diseases. And then the last one I want to go over is the influence for my drawing that I will present here in just a minute. And that is Staphylococcus aureus, a very common bacterial infection associated with sepsis, pneumonia, and skin infections. And it is increasing due to MRSA, and MRSA is an antibiotic-resistant strain of Staph aureus. That's what the SA stand for in MRSA. What I also found was interesting is that it actually contains surface adhesins to help stick to the airways, makes it even more difficult to get rid of. And these are my references right here for the two articles, as well as some of the images that I drew my inspiration from. So if you needed to look at the images to compare my drawings to their microscope images, then these are provided right here as well. But without further ado, we will look at my images here. All right, and then lastly, as promised, we'll look at my drawings here of the Staph aureus. We can see that well. So that first image here on the top, that is of a mouse lung that is uninfected. So if you see the two circles right about there, those represent the bronchioles, and then that represents one of the blood vessels here. This is unaffected, and this was taken, my inspiration was from a histology slide. And then right below here, this is of an infected mouse cell. Both of these are scaled to 100 micrometers. So this one is infected. These little pink strands here represent the Staph aureus. And you can see how much less defined it is compared to up top. Up top here, you're able to make out the bronchioles as well as the blood vessel. Here, you're not really able to make out a whole lot. It's definitely not as organized here. And then the second one I wanted to do, this was taken off of a skin cell here. That's this photo here on the right. The bacteria were magnified times 400 and the skin times 200. All these blue dots here represent Staph aureus infection as well. And then the brown represents the skin here. So you see a lot of them stick together here. Yeah. And then lastly, I also included a photo of a eukaryotic cell versus a bacterial cell. You can see here there's the nucleoid, which is where all of the genetic material is contained, followed with ribosomes. That is something that we find in both eukaryotic and prokaryotic cells. I also included a capsule, which some prokaryotic cells have, the plasma membrane, which is something that is shared also by eukaryotic and prokaryotic organisms, and then also a cell wall, which we will find in bacterial cells, that is of peptidoglycan, as we talked about earlier. And then also some of these contain fimbriae, and then also a flagellum for locomotion. And on the right here, this is our eukaryotic cell, one we're a little bit more familiar with probably, with the nucleolus and the nucleus. This is where our RNA is synthesized and genetic material is stored, contained by a nuclear envelope. We also have our endoplasmic reticulum. This one I drew as a rough ER, which has the ribosomes, and then this one I drew without the ribosomes. This also has the Golgi apparatus, mitochondria, and then the plasma membrane. So you notice that if we were to take a look between these two, this one is going to close by a membrane, and then this nucleolus here does not have a membrane. And you'll notice that there are a lot of closed membrane systems within the eukaryotic cell, where we do not find that within our prokaryotic cell here.